It's 11 o'clock, so let's go ahead and get started. We are actually coming toward the end of lecture exam 1 material and lecture. Remember the two biggest topics for points in lecture exam 1 we covered last lecture, and it's how our cells use the 3 nutrient links to make ATP. It's not making ATP from scratch, it's just putting the last phosphate group on ADP, and our cell will use ATP for energy. And remember that we started, of the 3 links, we started with glucose, a monosaccharide, we started there. And this is in contrast to catabolism that's done by your digestive tract. That's getting from chains to links. Once you get to single links, this is the catabolism done by cells. And it's going to take this one larger molecule, glucose, and split it into carbon dioxide and water. So that's catabolism still. But we built that framework first, and then we added fatty acids and glycerol in there, and then we added the amino acids in there as well. We have to take the amino group off, deamination, then we have the carbon chains, and we can just plug it into that framework. Any questions about that? Okay, now this is the next big topic for points on lecture exam 1, and I only have 2 lectures on this. And this has to do with DNA, which is a nucleic acid. Nucleic acids are biologically important. That's why they're listed as one of the 4 chains. It's just you don't have to eat them, so it's not one of the nutrients. We're going to talk about DNA basics first, the structure of DNA and what genes are. Then we have to cover RNA basics, and there's 3 different kinds. And then we're going to work through the sequence of protein synthesis. Now, this is the bare minimum of protein synthesis. I'm only going to cover the construction of primary protein structure. If you have your essays, go ahead and get your essays out. And you can see it's the first essay. We're only talking about getting primary protein structure. The other structures are a little bit more complicated. By the way, if you have that essay sheet out, you can see how, other than that first essay on the 4 levels of protein structure, everything else on that first page, essays 2, 3, 4, 5, and 6, were covered Monday. Glycolysis, Cori cycle, Krebs cycle, the chemiosmotic theory, and then ATP synthase. Remember, ATP synthase is attached to respiratory assembly on the matrix side. And when the protons are flying through the respiratory assemblies, it spins like a pinwheel and is a highly efficient transfer of energy, putting the last P on ADP. And that's why the vast majority of ATP come from that. Okay, so that's all the essays. This is going to have a lot of points on it as well. This will have some chemistry on some of the questions on lecture exam 1. This is basically it for the major portion of chemistry in the course, is this lecture exam 1. I try to get it all done in lecture exam 1 so we can kind of move on to human physiology. But these are all things you need to have from this class, so I got to cover it. All right, let's just go ahead and get to it. Any questions before we get started?
About the essays, is it depending on which version of the exam we get, we'll have what, 1 to 3 of them that we have to fill in? Or is every test going to have just all 6 we have to have entirely memorized?
No, everybody in this class are going to have the same essays, which was different from last semester. Okay, but it's going to be like 3 or 4 of them. And on Canvas exams and ERT, I'm not comfortable with having just things that can be copied and pasted. So a lot of this will be in fill in the blank form. instead of full-on paragraphs. It may be, like the short ones may be paragraphs, but, and you can also for ERT exams on Canvas, I'm not a stickler for complete sentences. Like if there's a question on the Krebs cycle, you can give me 3 bullets. I just want the content. Okay?
Okay, thank you.
All right. When I get back to Blue Books and live, then yeah, they're gonna have to write it out in complete sentences. All right. So this is where we left off. Again, we started with the framework of glucose going right down the center. And then I put in the glycerol and fatty acids on one side. And then I added lastly the amino acids. You got to take the amino group off, but the carbons, your liver can make those acetyl groups into just about anything. A lot of it goes straight into acetyl-CoA, but your liver can make that into pyruvate. Your liver can make that into the keto acids of the Krebs cycle. And this is one of the reasons why your liver is a top tier organ. It is just absolutely crucial to keeping you alive. All right, so let's talk about DNA, some DNA basics. Remember, nucleic acids are chains made up of nucleotide links, and to qualify as a nucleotide link, it must have 3 things. It must have a nitrogen-containing base, a sugar. In the case of DNA, it's deoxyribose. It has 1 less oxygen than ribose. So it doesn't have that exact formula of a H2O for every carbon. And then 1 of 4 nitrogen-containing bases. Now the phosphate group, again, is at least 1 phosphate group. You can have 2, or in the case of ATP, you can have 3. They're just called nucleotide triphosphates at that point. Where it says phosphate, this first diamond, you might want to write at least 1, which means 1, 2, or 3 still qualifies as a nucleotide. Okay, there are 4 nitrogen-containing bases in DNA. And you can tell right away that 2 are big and 2 are small. The big ones are double-ring purines. Adenine, which is the same nitrogen-containing base in ATP, is double-ringed. Guanine is also double-ringed, but slightly different in structure. The double-ringed nitrogen-containing base are referred to as purines. You need to know that for lecture exam 1. Cytosine and thymine are single-ringed, they're smaller. And those nitrogen-containing bases are called pyrimidines. I just remember here that the bigger structure is the smaller word. Now, for lecture exam 1, you do not have to know the exact chemical structure of adenine versus guanine or cytosine versus thymine. I will make you identify purines and pyrimidines. And it's easy. It's either double ringed or single ringed. Okay. So here's the difference between ribose and deoxyribose. Deoxyribose is just missing this oxygen. Okay, but here's the bases. We'll talk about uracil a little later. Okay, in our cells, DNA always exists as a double strand. It spirals in what we call a double helix. Okay. Each strand is held together by hydrogen bonds, which are weak. But there's a whole bunch of them holding the 2 strands together, which is why they pretty much stay together, unless caused to separate by an enzyme. Okay, now if you look carefully at these pictures, let's try to look at them with a critical eye. Just don't look at them passively, like you're you know, binging something on Netflix. Look at it with a critical eye. Notice that if we straighten out the double helix, and it looks like a ladder. Doesn't it look like a ladder right there? What are the vertical runners made up of?
Phosphate?
And?
Deoxyribose.
And the sugar. It is alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the vertical runners. What are the steps of the ladder made out of?
Guanine, cytosine, adenine, and thymine.
All the nitrogen bases. Now, what this is showing you here in the second bullet is to think of the double helix not like a biochemist. Think of it like a carpenter. The distance between the strands only allows for 3 rings, which is why a purine always matches up to a pyrimidine. A 2-ring purine must match up to a single ring pyrimidine because there's only room for 3 rings. That's the 20 angstroms. Okay? Now and if you think like a carpenter, okay, you got a big board and a small board to build the steps. One big board has 2 bolts. One small board has 2 bolt holes. The other big board has 3 bolts. The other small board has 3 bolt holes. And this is why the base pairing is highly, highly ordered and specific. A always goes to T with 2 hydrogen bonds. C always goes to G with 3. And that's what I wrote down here for the DNA nucleotides. A's always go to T's with 2 hydrogen bonds. G's always go to C with 3. Now, when you look at this, if you're looking at the double helix with a critical eye, are these 2 strand mirror images? Is there a left strand matching up to a right strand? You might see it better on this figure.
Yes.
Good guess. No, it's actually not true. Look at the sugars, since they are 5-sided pentagrams. This side, the pentagram's pointing up. This side, the pentagram's pointing down. What that means is that they're all left-handed. This strand is like this. The other strand is like that, upside down. And you can tell by where the phosphates hook up on the sugar. This phosphate is hooking up on the 5-carbon. That's why this is called the 5-end. This one's hooking up on the 3-carbon. That's why this is called the 3-end. Everybody see that? Another way to think of it is this is the head on the left side. This is the foot on the left side. But here the head is down here, and here the foot is up here. Just like what I did when I was younger, I'd be able to bend over a lot further. But you guys get my drift. It's all left-handed. This guy's this way, head is up. This guy's this way, head is down. But it's all left-handed. Okay. This is basic structure of DNA. It's from your textbook again, showing you this one is upside down. They're all left-handed. But C always goes to G with 3 hydrogen bonds. A always goes to T with 2. It's not a straight ladder. It actually coils into a double helix. Now, in our cells, it's never just DNA by itself. It's DNA and protein. And that's what makes up a chromosome. And the proteins are called histones, and they serve like spools, the DNA being the thread, and the DNA just wraps around these boulders like a spool, okay? In your regular somatic body cells, you have 46 of these threads. Now, this is a picture of what's called the human karyotype, which is a picture of the chromosomes at metaphase. Now, hopefully you got this from anatomy. This at metaphase is where the chromosomes are maximally condensed, so they have a consistent appearance. We take a picture of them, then we order them from biggest to smallest, the biggest being number 1. We go all the way to 22, and then the 23rd pair are the sex chromosomes. Now, on each of these chromosomes are genes. These are specific genes. Some of them code for proteins, which is our topic today. The code for the order of amino acids comes from the DNA genes with the order of nucleotide bases. Okay? But not all genes code for proteins. Some genes regulate, and that's the other topic. That's why I got to talk about protein synthesis and gene regulation. But there's a whole bunch of things here. Coagulation factors, those are the factors that are in the intrinsic pathway. Those are proteins that are coded for on chromosome number 1. Well, these two anyway. Got it. Do you know if you're Rh positive or negative? If you're Rh positive, you have this gene on chromosome number 1. And it codes for the D surface antigen on top of your red blood cells. I'm O positive, I have it. If you don't have it, that means both your chromosomes number 1 do not have this gene. You do not make the D antigen. That's why it's not on your red blood cells. Does this ring a bell? Hopefully something like that was mentioned in anatomy. But you can see that the genes for those proteins start on chromosomes. And this is a picture of just showing you the histone proteins acting like spools, and the DNA double helix is like a thread just wrapping around it. They just kind of lay really close to them during interphase, where you can't see individual chromosomes. But as the chromosomes condense for cell division, it coils and coils and coils over and over and over again. And then the chromosome is visible, and you can see it like this, and it looks like an X. All right. So let's just talk about genes now. And that's the topic here. Before the Human Genome Project, biologists thought even though there's only 46 chromosomes, they're huge. I mean, we're talking about 3 billion base pairs in those 46 chromosomes. And biologists thought there would be like 100,000 genes. And they did the sequencing and started backtracking to protein sequences. And they realized, oh, we're way off. And then they said, well, it's more like 40. We estimate 40 to 45,000. No, still way off. That's about 30,000. No, still way off. Now we're thinking it's more like around 20,000. That's all the genes. Well, out of 3 billion base pairs, that's less than 2%. What does that mean? That means most of our DNA is just space filler. Does nothing. Okay? Now that 2% is really important. Now this is crucial because every time your cells divide and these 3 billion base pairs have to duplicate, on average, on a sequence of replication, one of these 3 billion base pairs is misread on average. Well, as much as cell division that goes on in your body, that's why you don't ever have a problem with it, because the vast majority of those misread nucleotides are in space filler DNA. Okay, so if we're going to do protein synthesis, first we have to find that less than 2% that is a gene, and then that double helix has to unravel at that spot. It doesn't do it all at once. It might for really short proteins like ADH and oxytocin, but for longer proteins, it's just going to do it in a sequence. It's just going to start with the first part, and then it's going to start copying this messenger RNA off of it. And then the first part, after it's gone down the gene for a while, the gene actually starts closing off behind it. So it's only in the part there that it's being read does it unravel. And even on that, okay, only half of that double helix is actually the gene. The other half just sits off to the side. Now, even though it's not being read as the gene, I would not call it space filler, because every time your cells divide, you need that other strand that's just sitting off to the side to make the gene for the daughter cells. So it's not exactly space filler, but it's not being read. It's just sitting off here to the side. See this purple? It's just sitting off to the side. So only one side is the actual gene. Okay? Now what we have to talk about with these nucleotides in this long sequence of nucleotides, which is the gene, we actually read them in threes, what we call nucleotide triplets. Okay? And now we can start thinking about the sequence and protein synthesis. But before we do that, we are going to have to talk about RNA with a little bit more detail, because we really haven't covered RNA. But the bottom line, here's, okay, when you're supposed to take a step back and see the big picture, don't be intimidated, take a step back, see the big picture, what is this for? This topic is about going from a DNA nucleotide sequence to an RNA nucleotide sequence, and eventually to an amino acid sequence, which is primary protein structure. Everybody follow this right here. Put a big circle on this. This is the big picture of protein synthesis. It's about going from a DNA nucleotide sequence to an RNA nucleotide sequence, and then to a specific amino acid sequence, which is primary protein structure. What does it say on essay number 1? Describe the 4 levels of protein structure. Primary, amino acid sequence. Everybody see that? So we're going to start with DNA, and that's the gene or code. We got to copy it to mRNA. We'll talk about that in just a second. And that has the codon, and again, it's read in triplets. Codons are in threes. Then we got to go to an anticodon RNA sequence, which is on transfer RNA. But on the other side, so we have an anticodon on one side of the transfer RNA, on the other side is the amino acid. It's hooked onto the transfer RNA. Eventually, it breaks off the transfer RNA, and you get this growing amino acid sequence. This is the big picture of what we're studying today. Okay? Everybody got that? Does that help to have a big picture? All right, let's talk about RNA. And there's 3 different kinds. We just saw 2 of them just now. I'll go into more detail on those in just a second. First, let's talk about the differences of RNA to DNA. Obviously, the name is different. And since nucleic acids are named for the sugar, it's because the sugar is different. This sugar is ribose. It has one more oxygen. This is ribonucleic acid. Also in our cells, RNA is never double-stranded. It is always single-stranded. It'll form temporary hydrogen bonds with other single strands, including DNA. But it'll also form hydrogen bonds and nucleotides to itself, forming specific three-dimensional shapes. But it is, in fact, just a single strand. Third, there is one different nucleotide. Okay? So in RNA, we're going to connect that here with what I have on the whiteboard. There is no thymine. Instead, a very similar pyrimidine called uracil. It is very similar. This is an example of the second slide of the semester, specificity. Okay, it is very similar, but it's slightly different. And it replaces thymine in RNA. So in DNA, you have ATCG. In RNA, you have AUCG, with A, C, and G being the same. Okay? It's just in RNA, there are no T's. In place of T's, there's uracil, the U. It acts just like the T, thymine, forming two hydrogen bonds with the same adenine, A. And there's your three differences right there. Okay? The sugar, always single-stranded, and one of the pyrimidines is different. So again, it's ribose instead of deoxyribose, always in a single strand. And instead of thymine, it's uracil, but it takes the same place as thymine in DNA. And that is huge because people, here on the whiteboard, you have to know this complementary base pairing. A always goes to T, C always goes to G in DNA. A always goes to U, C always goes to G in RNA. And the opposite is true. T always goes to A, C always goes to G. U always goes to A, G always goes to C. The complementary base pairing. That's what the biologists call it, complementary base pairing. Okay. Now let's talk about the three kinds of RNA, and then we can get to protein synthesis. Messenger RNA is the product of the first step of the sequence we call transcription. And again, A goes to U, C goes to G. And whenever I would write this down as a student, I would always write it down like this, so that I'm always reminded which one has 2 hydrogen bonds, which one has 3. Transfer RNA is that molecule, that RNA that has the anticodon on one side and the amino acid on the other side. In 3 dimensions, it looks like this. Anticodon is down on the bottom, up on top. In this particular case, is aspartate, which is an amino acid, aspartic acid. Remember that from last lecture, lactate and lactic acid. But we are going to need some ribosomes, the organelles for protein synthesis, and that's where our RNA comes from. In fact, this is the best illustration of going up from macromolecules to organelles. A ribosome, an organelle, is just two subunits, a large and a small, made up of ribosomal RNA, rRNA, and protein. I mean, it's pretty big. There's actually 4 single strands of rRNA and 82 proteins that make up a ribosome. So it's huge, but it's still just macromolecules. So those are the 3 forms, and we need all 3 to do protein synthesis. Okay, so the gene is copied on messenger RNA, but it is complementary RNA nucleotides. Transfer RNA has, again, you can see that it's a single strand, but it will form those double bonds in sections, forming loops. Well, the anticodon is on loop 2, and the amino acid's on the other end. And here's the 2 subunits, one large, one small. They have 2 binding sites on them that fit transfer RNAs. We'll talk about that later. And I don't know why they call it this, but the first one is the P site, and the second one is the A site. P and A. All right, so here's the overall figure for protein synthesis. If you have your lecture packets out, these are the pages that are relevant to what we're talking about today. I'll expand on some other things next week. We only have one more lecture of lecture exam 1 material, people. All right, so on page 13 is again some definitions of DNA and RNA, and looking at some words that we are going to explore in more detail, like codons. We already talked about genes. We just about to talk about protein synthesis. Page 14 has the chemical formulas. Again, I'm not going to make you memorize biochemical formulas. This is not a class on biochemistry, but you should be able to identify purines and pyrimidines. Oh, one other thing. You know, I'm glad I just remembered this. On the very early slide showing you alternating sugar phosphates. This is called a phosphodiester bond. And it's the phosphate between 2 sugars. You do have to visually recognize that as well. I did tell you you needed to visually identify peptide bonds. And that's just between NCC to the next NCC. That's in proteins. Well, this you're going to have to visually recognize phosphodiester bonds. And that is between sugar and phosphate. And a sugar could be deoxyribose. It can also be ribose. That's still a phosphodiester bond. Okay? Now this figure here from the PowerPoint is the equivalent to page 15 in your lecture packet. It's a little bit more of a simplified version, this figure. But we are going to start with the gene, which means we're in the nucleus. That's where the DNA is. The first step is transcription. The second step is translation. That's happening out in the cytoplasm. Transcription in the nucleus, translation in the cytoplasm. Everybody got that? Okay. Any questions so far? This is kind of like all the preliminary stuff before we get to the meat and potatoes of protein synthesis. You do have to know the structure of DNA and RNA and the differences between DNA and RNA. because you're going to see all three forms of RNA in protein synthesis. Okay, let's start over here. All right, so we're going to start in the nucleus. And there's an enzyme in the nucleus called RNA polymerase. Let's see that in purple. So there's actually another enzyme called helicase that unravels the double helix at the point of the gene. But the code is the DNA. And in the nucleus, this enzyme, after it unravels, is going to start making a complementary strand with RNA nucleotides. This is the messenger RNA. So watch this animation. It's deoxyribose in red down there. So that's the DNA gene on the bottom. And you got ribose there in kind of brown. Those are RNA nucleotides. Notice how each nucleotide is a triphosphate. They get those phosphate, high energy phosphate groups from ATP. Each nucleotide is requiring an ATP. Okay, now watch the animation because I want you to get this. Notice that the hydrogen bonds form first and then the phosphodiester bond. Hydrogen bonds first, boom. Phosphodiester bond second, boom. Everybody see that? This is powered by those triphosphate nucleotides. That's ATP right there. But hydrogen bonds first. So you got to have the right number of bolts and the right size board to get an exact complementary strand. But remember, in RNA, there's no T's, that's why there's U's. If this was DNA, A would be going to T. But since this is an RNA up here, A has to go to U. Because in lecture exam 1, I can give you a strand of DNA, and you better give me the correct RNA complementary strand. That will be expected of you on lecture exam 1. Okay? So this is transcription, and it's happening in the nucleus. So we're going to go from nucleotide sequence to nucleotide sequence. It is from DNA to RNA. But since it's still a nucleotide sequence, we call this transcription. Okay? It's done by this enzyme, RNA polymerase, and it's in the nucleus. Now we're going to eventually get to this next step called translation. And the way I remember this, and it's very easy to do so, is if you think about what happens in court. In court, you have court transcribers doing transcription. And they're writing down spoken English into written English. It's still English. We are going from a nucleotide sequence, DNA, to a nucleotide sequence, RNA, but it's still a nucleotide sequence, transcription. Does that help at all? Because now we're going to go from a nucleotide sequence to an amino acid sequence. And that's like going from English to Japanese. That's where you need a translator. That's translation. Does that help at all? Okay. All right, let's go on here. When there is a terminator sequence, it causes RNA polymerase to fall off, transcription stops, the single strand messenger RNA dislodges and exits the nucleus because it's got to go out to the cytoplasm. And that, if you were ever curious, is why there is nuclear pores in the nuclear envelope, because messenger RNA is a big molecule. And if you have pores in the nuclear envelope, you can get messenger RNA out a lot easier. Does that make sense? All right, now it's out in the cytoplasm. Oh, this is showing you a figure here. Here's that RNA polymerase. It separates. There's actually a separate enzyme that causes the unraveling of the double helix. But only one of the strands is red. The other one just sits off to the side. They call it the antisense strand, but don't worry about that. And it's a complementary base pair. Now, once we get out into the cytoplasm, this translation is going to happen in 3 steps. And this is now out in the cytoplasm. First step is initiation. And that's why this was not random letters up here. Remember that codons are always on messenger RNA. This is the code or gene on DNA. This codon is on messenger RNA. Anticodons are on transfer RNA. Got to have that straight. And the initiator codon is AUG. That's the first step, initiation. I'm just going to put a number there. Initiation. Now AUG, the codon, will match to an anticodon with its UAC, and that anticodon, the amino acid is methionine. And methionine, in the initial construction of a protein, is always the first amino acid. Now here's, you can see in this figure, the P site and the A site. P is first, A is second. Okay, we'll go over this in just a second. AUG is the initiator codon. This animation, when I replay it, is going to show you elongation. That RNA or ribosome is going to settle in on that messenger RNA, and the P site and the A site is like this. Well, methionine is going to go into the P site, then whatever matches this codon with this anticodon, I don't have it memorized, but that's going to be amino acid 2. That goes on the A site. Okay? Now, before it slides, and that's all the ribosome is going to do, this big orange blob here is the ribosome with rRNA. So all 3 forms of RNA are involved: messenger, transfer, ribosomal. Then what it's going to do is it's just going to slide down the messenger RNA. Remember, it's clamped on this guy. So the small subunit and the large subunit clamps on this guy, and it's about this big. But it has in its side, so I'm not going to draw it, but the ribosome blob would go all the way over here to the messenger RNA. Everybody got it? I'm just going to stop it right here. Well, once that second amino acid occupies that spot, it slides down the messenger RNA. Now this is the P site. But before it does that, it breaks this covalent bond and forms this covalent bond. And this is the peptide bond. The first one. So now amino acid number 2 is on the P site. A site is open. Well, if this is the codon, the transfer RNA with this anticodon for amino acid number 3 now occupies the A site. And before the ribosome shifts again, with this being the P site, this covalent bond is broken and a new one is formed. Boom. Now the shift in the ribosome now puts amino acid number 3 from the A site to the P site. Now you can add the fourth one, and on and on and on. You do this 9 times, you get oxytocin. You do this 280 times approximately, you get one of the globs of hemoglobin. Does this make sense? But notice the ratio of amino acids to nucleotides is 3 to 1. And that's because of the transfer RNA. Nucleotides to amino acids is 3 to 1. Does everybody see that? All right, let's round up the beta chain of hemoglobin to 300. It's like 278, something like that. But 300 amino acids. How many ATPs does it take to make a 300 amino acid beta chain?
900.
900. Because go back to that animation. Each one of those is a triphosphate. It's got to get those from ATP. So if you have to make 900 nucleotides in your messenger RNA, that's 900 ATP just to make a beta chain. And there are 2 beta chains and 2 alpha chains in hemoglobin. And that's just one hemoglobin. That's just one quaternary hemoglobin. Do you see how much energy it takes to do anabolism? Because what we are talking about here in making a protein is anabolism. So this anabolism is done by cells. To make 300 peptide bonds, you need 900 ATPs. That's the energy in to build up a protein. Does that help at all, seeing that, connecting those dots? It takes energy to build up large chains. This is what we're doing with protein synthesis, is doing anabolism. And if you look on page 12 of your lecture packet, those are all the anabolic reactions. I don't cover too much of that in lecture exam 1. But under proteins, number 2, those are dehydration reactions that get you proteins. And you got to put a lot of energy into it. Okay? So that's the second. By the way, I'm going to be doing my videos for Zoom meetings differently starting next week. I'm going through all this training on this new equipment. Here, I'll show you. Got this camera in the middle. It's going to have presets on it. I'm going to see you on this huge flat screen TV. This is the, I'm usually behind this desk looking out. Well now, and then I'm usually looking at this tiny screen on my laptop. Now I'll be looking at you on this big screen. And using this camera that has 3 presets. So instead of rolling this projector cart, I'm just going to push a button, and supposedly you will get a little torqued aspect ratio. So, you know, it's kind of like, look at this left board from here. See how it's kind of torqued because it's diagonal? This camera actually corrects for some of the aspect ratios. And all I have to do is push a button. And I don't have to roll this cart, which right now has all these cords, and that's why it's going boom, 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 because I'm having to roll the cart over the cords for this new equipment. So hopefully it'll be a little bit smoother next week. And I looked at the camera on Monday. It is far superior than this webcam I'm using. So hopefully you will appreciate the efforts, because I'm sick of learning curves, by the way. I've had like 200 learning curves since ERT. So this shifting of the ribosome 3 nucleotides at a time and adding a peptide bond, that is elongation. And it is a 3-to-1 ratio, nucleotides to amino acids. Okay? This one is saying that the anticodon is complementary to the codon. Okay? Another way of thinking about complementary base pairing is it's the opposite or negative. You following me? If you're following the A to T, C to G, or A to U, C to G, it's like a negative. So what is a negative of a negative? It's a positive. So look at this 3rd amino acid. Right here is the 3 DNA nucleotides in the gene, CGC. What does it become in the anticodon? GCG. Well, what is it in the anticodon? CGC. This CGC is exactly the same sequence because it's a negative of a negative. Okay? You got to follow this. There are 2 C's in this, 2 G's in this, 2 C's in this again. Negative of a negative. Does everybody see that on the whiteboard? Okay. And then the 3rd step is finally you got to get one of 3 terminator codons. And it goes all the way back to the gene, and you just have to go complementary base pairing. There's actually a terminator triplet on the gene. It would be on this one, ATT. If there's an ATT triplet, boom, everything stops, RNA polymerase falls off. Or it could be, and I'm just doing this on the fly, ATC. And I can't see the last one because your pictures are in the way. Or it could be ACT. Any of those 3 on the DNA code will get you this terminator codons, UAA, UAG, or UGA. You do have to know the initiator codon, and there's only 1, and the 3 terminator codons for lecture exam 1. And when that happens, the 2 subunits of the ribosome separate. Messenger RNA floats off to be digested, broken down into individual nucleotides and recycled, and everything stops. So 3 would be termination. And again, you had over here one of those 3 terminator codons. Just remember, codons are on messenger RNA. Okay? But now we're going to go from a nucleotide sequence to an amino acid sequence. This is all in the cytoplasm. That's like a different language. That's translation. But translation is in 3 steps. Initiation, I should probably get on the camera here. Translation is in 3 steps. Initiation with the initiator codon, elongation with the ribosome sliding 3 nucleotides and adding a new peptide bond. And finally, one of the terminator codons is going to get everything to just release. So that's termination. That are the 3 sub steps of translation. Does this help at all? Nucleotide sequence to nucleotide sequence, still nucleotide sequence, transcription. Nucleotide sequence to amino acid sequence, different language, translation. Okay, I'm going to leave that up there so you can see some of the amino acids still stuck to the transfer RNA.
When you go back to the PowerPoint, can you restart the elongation GIF? I missed that.
Okay. Okay, here's initiation. Methionine is the first amino acid. Those are the 2 yellow blobs of the ribosome. Now watch it just shift 3 nucleotides over. You can see in red the covalent bond with the transfer RNA breaking and the new peptide bond forming. Until you hit one of the terminator codons, you get a releasing factor and everything falls apart. In this case, it was UGA.
Thank you.
Are those 3 terminator codons the kind of like the ones we'll most see? Or are they the only ones we'll see?
Those are the only ones you'll see.
Okay.
Okay. This is on the whiteboard. I said we're going to look at DNA basics. We covered that. RNA basics. We covered that. Protein synthesis sequence. We just covered that. Any questions? This is figures from your textbook. And again, it's showing you the P site and the A site. P site first. This is initiation. Then the second one will come in this case it was serine that occupies the A site. Once both of them are occupied, the covalent bond for methionine breaks. A new covalent bond called a peptide bond forms between the first 2 links. Once that forms, the ribosome shifts 3 nucleotides. Serine, which was in the A site, is now in the P site. Now there's room for glutamate. This is going to be the 3rd amino acid. It's going to, before it shifts again, it's going to break this bond, form a new peptide bond. Then it shifts 3 nucleotides so that glutamate, instead of being in the A site, is now in the P site. That is elongation. Okay? Now, in our cells, there are free floating ribosomes in the cytoplasm that will clamp onto any messenger RNA. If the messenger RNA is long, you can have several ribosomes clamp on it. We call that a polyribosome. This is an extremely high magnification. Extremely high magnification. We're almost being able to see biochemicals here. Horizontal line is the messenger RNA. These little caterpillars coming off are growing peptides. And the big beads, well, those are the 82 proteins and 4 single-stranded ribosomal RNAs of ribosomes. This is an electron micrograph of extreme magnification. We are actually seeing biochemicals. But if a cell has to make a lot of proteins, it's not going to bother with even a polyribosome. It's going to have a whole bunch of rough endoplasmic reticulum in it. It's rough because it's covered with ribosomes. And that sets up a space-efficient assembly line for making a ton of proteins. So like beta cells, in your pancreatic islets that are making insulin, or even your alpha cells making glucagon. They're loaded with rough endoplasmic reticulum because those hormones are proteins. Cells in your red bone marrow, they've got to make red blood cells, erythroblasts, loaded with rough endoplasmic reticulum because it's got to make a ton of hemoglobin. Okay? Now, it's much more complicated than this. With cells that make a lot of proteins, there'll be rough endoplasmic reticulum that'll bubble off little vesicles. We call it transport vesicles of this preliminary protein. Most proteins actually come off this way, not all, but most. And the reason why is this. This AUG codes for methionine. Very few proteins actually have methionine as the first protein in the final product because it's going to go through the Golgi complex and it's going to be refined to the functional protein. And oftentimes when you finally get to that functional protein, methionine is gone. Does this make sense? We will look later. Actually, we will see this next week in lab 3. So let me connect this with lab 3. 3B is done with pepsin. Pepsin is created in some of your secretory cells in the stomach. But those cells cannot make pepsin. Why? Pepsin breaks up proteins. And if a cell makes pepsin, it would digest itself. So it makes pepsinogen, which does nothing. And pepsinogen has as its first amino acid methionine. Well, then the cell just spits it out to the stomach's lumen. It's exposed to stomach acid, and the first 44 amino acids fall off. Bye-bye methionine, it's gone. It's not the first amino acid of pepsin, but it was of pepsinogen. Does everybody get that? So even though methionine is always the first amino acid, it is rarely the first amino acid of the final protein. This is, again, an amazing magnification. 0.5 microns, that's tiny. And this is showing you here. So like, if this was a beta cell, what's actually being made is proinsulin, does nothing. Goes to the Golgi apparatus, gets a disulfide bond, breaks this bond over here, a couple hydrogen bonds are added, and boom, functional insulin. That's put in a secretory vesicle that goes through exocytosis every time you have a Snickers bar. Okay? Methionine is the first amino acid here. Methionine is not the first amino acid here. All right, last topic for today is gene regulation. And this will be pretty quick. This topic is discussed on page 17. On page 16, there is a picture on the top half of page 16 on transcription. We will compare it to what I'm going to cover on Monday. But all we've covered so far on page 16 is the top half. Now this topic of gene regulation is on page 17. There are 2 forms, and you need to know both forms, both of these forms on lecture exam 1. There's many other forms of gene regulation. This is a complex topic, but I'm just going to leave it very simple. You have to know positive control, and the example I give you of positive gene regulation is induction. Positive in that the gene is normally off. When you introduce an activator, you turn it on. Positive, on. It's off, you turn it on. You induce it to turn on. That's why it's called induction. Okay? In its most basic form, what it's doing is it's bending the DNA double helix to be in a much more favorable shape for RNA polymerase. Positive control, turning on. You can see it's actually quite a bit more complex than what I'm giving you. Shall we keep it at the simplest level? Okay. The other one is negative control. The example is repression. In this case, you actually have greater control. The gene is actually normally on, but if you introduce a repressor, you turn it off. Since you're going from on to off, this is called negative control or repression. Now, these are some of the genes, some of the 20,000 genes that don't code for proteins. These are genes, though. Promoter gene, operator gene. What is being transcribed is the region in red. Excuse me, in green. This region in green is what's going through transcription. Okay? Well, the repressor blocks RNA polymerase. That's how it turns it off. But the reason why this has more control is you could just go ahead and remove the repressor or put it back. Then remove the repressor and put it back. So in this form, you can turn it off and on and off and on and off and on relatively easy. And this is the basis for the lac operon gene regulation. And this is what is described on page 17. Now, I've given you a lot of content this week, haven't I? See all these heads nodding. So I'm going to make it easy on you. For lecture exam 1, you just need to know this slide and the previous slide and know that the lac operon example is an example of this, repression. Okay? Now I'm going to walk you through this because you really should be exposed to this. And the reason why we're doing this lac operon is because it is simple. It's simple because it's done in bacteria, and bacteria gene regulation is way simpler than in our cells. In our cells, it's way more complicated than this. But you got to be introduced to the facts of your genes are controlled. They're not always just going on. They're going on when they're supposed to go on. And so I want to use this thing, since you know there's not going to be any specific questions on this, except for this is an example of negative control repression. That's all you got to know, okay? But I'm going to use it as an example of how you take a complex concept that you have to learn that can seem to be intimidating, and how you take a step back, see what the big picture, what it's for, and then break it up into small sequential steps. And before you know it, you're thinking this is a piece of cake. Are you ready? The big picture, as I take a step back and see what this is for, is this. An E. coli bacteria does not want to waste ATP making a protein it doesn't need. An E. coli and your cells have this in similarity. An E. coli does not want to waste ATP making a protein it doesn't need. If you got that down, the rest of the sequence is easy. Okay? Now, one of the proteins is an enzyme that's made off of this gene. There's actually 3 genes. So they call the genes that are actually getting transcribed structural genes. Well, one of the proteins is this beta-galactosidase, and it is used to digest lactose. All right, so normally the operator gene has a repressor molecule stuck on the DNA that blocks RNA polymerase. So here's the repressor, it's on the operator gene. RNA polymerase can't come in here to make messenger RNA. It's blocked, okay? And the reason why there are 3 proteins made right in a row is because bacteria have a single, well, E. coli has a single ring DNA as its one chromosome. Very similar to mitochondria. Mitochondrial DNA is a single ring of DNA, which is why they think it was an ancient bacteria. All right, well, if lactose passively enters the cell, the repressor protein actually has binding sites for lactose. And when lactose binds to the repressor protein, it changes shape and causes it to fall off the operator gene. Well, now the DNA is no longer blocked from RNA polymerase. It can move on in and start doing transcription. And it does it all three in one, and one of them is beta-galactosidase. It is burning up a lot of ATP to make this. But here's the deal. It can now digest lactose and make even more ATP from the food source. It was willing to spend ATP to make a digestive enzyme so that it can make even more ATP. It invests some, gets a big payoff. Okay? But when the last lactose is digested, now it's going to fall off the repressor protein because they're all gone. So it could digest the last lactose, the last ones that were stuck on the repressor protein. Now the repressor protein gets back to its original shape, which has a high affinity for the operon or the operator gene, and it sits back down on the DNA and it blocks it and it turns it off. So the only time this E. coli expended ATP to make this protein is when there was lactose, because an E. coli does not want to waste ATP making a protein it doesn't need. The big picture. Okay. Now I do need you to know this slide too, though. So you just need to know that the lac operon is an example of repression, negative control. Was that helpful for me going through that step? There are going to be things in your future that you're going to look at me going, oh, this is going to kick my ass. Don't be intimidated. Take a step back and figure out what the whole thing is for. That makes sense of the whole thing. Break it up into small sequential steps, and it now makes sense. The reason why I want to bring this up to you is because of the cyclic AMP. This E. coli can make even more ATP if it had glucose instead of lactose. All right, and so if there is lactose present, but glucose is also present, this cyclic AMP, or little c AMP, remains low. But if there's no glucose, and it's not going to get the even greater ATP yield from glucose, well then, okay, I'm going to try to digest the lactose. Cyclic AMP levels rise, and it's going to maximize beta-galactosidase. This is just add one little layer of complexity, but since I'm not going to ask you any specific questions on the lac operon, you should be able to have it. What is cyclic AMP? Well, it's this. If you have adenine, ribose, and 3 phosphate groups, what is that? Because you also have to recognize this structure for lecture exam 1 as well. What is this? Oh, I better. What is this?
ATP.
ATP. Piece of cake. What is this?
ADP.
ADP. What is this?
AMP.
AMP, adenosine monophosphate. But if you put it through reaction by this one enzyme called adenylate cyclase, it does this. In AMP, there's only one covalent bond between the phosphate group and the ribose. But in this, adenylate cyclase will make the phosphate group bond twice to the phosphate, forming a ring of bonds. That is a circle of bonds. And this is cyclic AMP. And the reason why I want you to know it is we'll see it again during the semester. We have hormones that signal throughout the whole body. We have neurotransmitters that go to a really short distance. Well, cyclic AMP is an intracellular messenger. And we'll see it again later in the semester. Intra, within the cell. And that's what it's referring to here as a signaling ligand. It is messaging within the cell. And in the E. coli, it's saying, let's try to use glucose if we can. Oh, there's no glucose, only lactose. Okay, well then, let's rise the signaling molecules to say we better maximize lactose. All right? And there's some other things if you want to look them up. Again, I'm not going to ask very many questions on, well, I'm not going to ask any specific questions on lac operon, except that it is an example of negative control repression, and that the E. coli will also use cyclic AMP to control which food source it's going to use. All right, now we got a little time before lecture exam 1, and that's because I'm going to hold off until President's Day holiday to have that lecture exam. But we have one more lecture on lecture exam 1 on Monday, and then we're done with lecture exam 1 material. Okay? So let me go over the whiteboard. And we got done in time today. So that's showing you cyclic AMP, intracellular signaling ligand. You can just call it a messenger. Oh, this will be so much better when I don't have to go over these chords. That's showing you protein synthesis, the sequence. First you got transcription, and then you got translation. Translation has 3 subphases: initiation, elongation, termination. That's the sequence. Transcription's in the nucleus, translation's in the cytoplasm. And this is the left side. Tying it all together with the 4 biologically important macromolecules. Because the 2 biggest sources of questions for lecture exam 1 is right here. First for ATP and how we use the 3 nutrient links to make it. And then now it's going to be on DNA. First, and what we covered today was how DNA codes for proteins. And then on Monday, we'll talk about how DNA codes to make itself and DNA replication. All right. Any questions? All right, Wednesday lab, I'll see you in about 40 minutes.
I have a question, Professor.
Okay.
I was wondering if you could give an example of the gene regulation in the human body.
There are very similar mechanisms in our digestive tract for controlling the digestive enzymes. For example, if you eat a real fatty meal, you'll get a higher amount of both bile salts and lipase enzymes coming out of the pancreas, as opposed to if you ate like high vegetable, high fiber meal. If you had a high vegetable, high fiber meal, you would have different digestive enzymes. You would still get all of them. It just will adjust the ratio of enzymes it's going to make and release for the same reason. Your body doesn't want to waste making digestive enzymes, proteins. If it's not, you're not going to find that food there. Does that make sense?
Yes, thank you.
Okay. Any other questions? All right, Wednesday lab, I'll see you in about 40 minutes.